Hello, welcome back. Good to see you all again. We're now going to look at our next AS revision lesson, which is the biodiversity, which is the final topic in the AS AQA syllabus. So the first thing I want to look at when we're looking at biodiversity is the concept of what a species is. So it's a difficult one for scientists to develop this idea, and there is no 100% perfect definition because there's always a few exceptions to prove the rule. But the primary one that we use is that a species is a group of similar individuals that is capable of interbreeding and producing fertile offspring. So make sure you get that in your answers. So the idea that you've got similar individuals, they can interbreed and produce fertile offspring. So you might find in some cases that some species might appear to be similar in appearance. So like a tiger and a lion. Now I appreciate their coat colour is quite different, but if you gave me two skeletons, one of a tiger and one of a lion, I could not tell the difference. They are very, very similar in their physical proportions and build and skeletal structure and muscle structure and organs. It's only really the coats that look a bit different in their behaviour. So yeah, okay, they might be the same species because they're similar, physically. They can actually interbreed. They will actually mate with each other in captivity. So already to tick two things off, so maybe a lion and a tiger are the same species. But when you breed the two together, you end up producing offspring that are infertile. So ligers, which are the hybrids between lions and tigers, are infertile. So they cannot reproduce themselves. The reason for that sterility is because the offspring have an uneven number of chromosomes. So as a result, they cannot form bivalent, so it's homologous pairs in meiosis, and therefore they cannot make their own gametes. So we therefore know, by applying that species concept, that lions and tigers are separate species. So that's the beginning point. We'll stick with this idea of species and classification as we go forward. So a very famous Swedish scientist, Carl Linnaeus, came up with the classification system that we still use today, and he came up with what we call the binomial classification system. So giving every species on the planet two names. The first name is the genus, the generic name, followed by the species name. Now, by having this sort of binomial system, it shows you that the relationships between species, because we can identify that, oh yes, they're in the same genus, so therefore they must be closely related. And then obviously we've got the specific name that distinguishes that group from other members of that genus. So I'll write out a specific binomial name of an organism. Hopefully this is one that you've all heard of. It's one of my favourites. Tyrannosaurus rex. There we go, T-Rex. So you'll notice how I've written this is not in any old way. I've written the genus name first, so Tyrannosaurus, followed by the species name, Rex. And we always write the genus name with the first letter capitalised and the first letter on the species name lowercase. So that's Tyrannosaurus rex. That's its scientific name. That's its binomial name. So we've only really encountered two levels of classification so far, and that is the genus and the species. There are a wider range. There are many more levels of taxonomy, which is the sort of the study of the classification that we can look at. So I'm going to write out those letters. I tend to remember this as a mnemonic. There are many good mnemonics out there that you can use. I certainly would recommend coming up with your own. I still use one that me and my friend came up with when we were at A-level. So we'll have a look at that taxonomic levels there next. So like I said before, in the classification system there are many levels. It's a hierarchical grouping. So what hierarchy means is groups within groups with no overlap. A bit like the army, if you like. If your lowest ranking sort of privates, corporal, sergeant and so forth going up, they're all within this larger grouping, but they represent smaller groups and they don't directly overlap. So how do I remember it? How do I remember this one? So one that I remember from A-level myself is, did Karen prey cunningly on fat green spiders? There are many others. I can't necessarily repeat all of them on this video, but another one that one of my other A-level students in the past came up with is, did King Philip come over for good soup? Another one you could use there. So what do these letters actually stand for? Well, the first letter stands for the domain. This is the largest group there is in the classification system, and there are actually three main domains that we need to look at, and they are the prokaryotes, the bacteria, the archaea, and the eukaryotes. Then the next level down is kingdom. You need to be aware of the eukaryotic kingdoms. There are the animals, the plants, the fungi, and the protists, or the proterozoa. In reality, if you ever do degree level biology, you'll realise actually there are way more kingdoms in the eukaryote. There's something like 60 or 70. The proterozoa is a bit of a dustbin kind of group, but actually you could subdivide that significantly into many different kingdoms. But for A-level, we stick with animals, plants, fungi, and protists. The next level down is the phylum. Then we're followed by the class, the order, and the family, the genus, and the species there. So you need to be able to know the correct order of these different taxonomic groups, and often you see exam questions where you're filling in the missing words and so forth. So be aware of that particular list. Not always they have domain on there, so be mindful of that. You might be starting off at the kingdom level. Look at what other bit of information. If it's animalia or animals, then you know we're looking at kingdom. If it's domain, it'd be looking at terms like eukaryota, bacteria, or archaea there. So let's look at an example. Let's look at us as a species. So the domain we're in is the eukaryota. The kingdom we are in is the animalia, the animals. The phylum we're in is the chordates, the vertebrates, animals with a rigid notochord running down the body. Ours is ossified with bone as well. The class we're in is mammals, so the vertebrates that have hair, produce milk, typically give birth to live offspring, that sort of thing. The order we're in is the primates, so we share this with monkeys, apes, lemurs, tarsiers, that sort of thing. The family we're in is the Hominidae, so the apes, the great apes, which includes chimpanzees, gorillas, orangutans, and us, and some of our extinct relatives. So that's our family. The genus we're in is Homo. So we're actually the only living members of our genus today, but there were several species that have gone extinct, like Homo neanderthalis, Homo erectus, Homo floresiensis, etc. And obviously the species for us is sapien. So our binomial is Homo sapien, which means wise man, which I can sometimes be debatable with our species, but that's what we are. So there's a nice little example there. This obviously applies to any organisms that have been classified. We can do it with plants, we can do it with all sorts. So you need to be aware of that list, that order, and be able to apply it. So the next thing we're going to look at is, how do we make sure that species will, or individuals within a species will mate with their own species? And so we're going to look at courtship behavior. So a courtship behavior, it plays many important roles to ensure that we don't get hybridization, so different species interbreeding, because hybridization for many species is quite disastrous, because you're going to put a lot of time and energy in producing offspring that they themselves cannot reproduce. So therefore they're not passing on your alleles to the next generation, which is quite disastrous in natural selection. So natural selection and sexual selection has driven species to carry out specific courtship behaviours to ensure that you are going to definitely reproduce with the same members of your species. So courtship behaviours tend to be stereotypical within a species. They always follow a very kind of regimented pattern that's quite distinctive. So certain courtship behaviours you might have noticed is, I often see in the garden, is wood pigeons. So you'll see a male wood pigeons always seem to be harassing female wood pigeons, and they do this very stereotypical courtship behaviour. I'm going to do a little impression of it now so you get the idea. And you'll see them, they'll walk up to the female, they puff out their chest, their throat, make it really big and obvious. It's actually quite brightly coloured, the throat, if you look quite closely. So that probably gives them this sort of visual signal to the female, look, I'm interested. And then they start to coo, and then they start to bow like this, and you see the head going down and the tail cocking up at the back there, all the while they're puffed out and cooing away. And now if the female's interested, she will mate with the male, or often I see that she just takes one look and flies off. Now, that courtship behaviour is quite important because the male and female are, probably assessing whether, firstly, they are both, receptive and able to mate. Courtship behaviour also might increase the chances of fertilisation by maybe triggering gamete release and so forth like that. It can help form pair bonds as well, which might make rearing offspring more successful because you're a tight unit, you're going to work together. And there are a few other reasons for why courtship behaviour is important. And I would always recommend that you have at least 2 or 3 specific reasons for why courtship behaviour is important. I'm just going to write them on the board now, and you can always jot them down in your notes later. Okay, so there's a couple of last little bits to look at with the taxonomy idea. I alluded to the idea of the 3 domains of life, and this was recognised by another scientist called Carl Woese. And he realised there was a group of what everybody considered to be bacteria for a very long time, often the extremophile bacteria, so bacteria that live in very extreme conditions, so very high temperatures, high pressures, high salinity, that sort of thing. Everybody assumed they were bacteria, but he looked a little bit deeper. He looked at obviously their cell structure, but also their DNA and also their molecular biology, and found out actually that these group of bacteria, which he called the archaea, were actually a completely separate domain of life, independent of the bacteria. They probably share a common ancestor, but they are very, very different. So what makes archaea different to bacteria is that they have ether bonds instead of ester bonds in their cell membranes. That's quite a big difference there in terms of their lipids. They have a more complex form of DNA polymerase, so maybe a little bit more like eukaryotes in that respect. Their DNA is associated with proteins, where in bacteria it's not. So in some ways I sort of see them almost kind of a little bit like a transition between the bacteria and the eukaryotes, because they seem to have a bit of features of both. So bear that in mind. You need to have a few of those key features when you're looking at archaea and what makes them a bit different, maybe like a 2 marker or something like that. The last little bit to look at here with the taxonomy is the idea of phylogeny. So phylogeny is the study of the evolutionary relationships between organisms. So how do different groups, how do different genera, families, orders, classes, how are they related? How do they fit together? And through this sort of phylogeny, phylogenetic studies, we've been able to build up what's called the tree of life. So how every species on the planet is actually interrelated. We're all related to every organism on the planet, from the tiniest bacterium to the mightiest oak tree. We're all actually related. Even I'm related to a banana plant, believe it or not. But we do share quite a distant common ancestor. So what you might see with these phylogenies is so-called phylogenetic trees. I was going to draw a very quick one of the chordates, the animals with backbones. So we've generally got our sort of bony fish down here. Now there actually are other groups of fish that I could branched off earlier, like the jawless fish and the cartilaginous fish. But I'll start with them. So we've got the bony fish here, and the bony fish, things like salmon, trout, pike, that sort of thing, still exist today and they're doing quite well. But one group of these actually split off and gave rise to all the animals the vertebrates that live on land, the so-called tetrapods. So animals with 4 limbs, any 4-limb vertebrate we consider to be tetrapods. So that's a character state that would separate off these groups from the rest of the bony fish. And the first group here are, of course, the amphibians that we're all familiar with. I'm just going to write A there because I'm running out of room. So they're the amphibians. They themselves gave rise to the reptiles, which I'll put as an R here. And the reptiles diverged off into a wide variety of different groups. Some gave rise to the mammals, others gave rise to the birds, and of course we also have our modern reptiles as well. So you can see we've got a sort of phylogenetic tree. that is sort of developing from this. And we can see that, you know, birds are actually more closely related to other reptiles, mammals are kind of a bit more distant there, and obviously all three of these groups, reptiles, birds and mammals, are more distant related to the amphibians because they share a more distal common ancestor. The closer the nodes are, i.e. the branches, the more closely related they are, the more distant they are, the less closely related they are. And I could quickly show you on maybe a better diagram than the one I've just drawn. You know, something like this might be a better example. So you might have, these represent different species, and we can see that A and B are more closely related because they share a more close common ancestor, whereas A and B and C, their common ancestor is down here. So that, and obviously the longer these lines are, the more kind of geological time has taken place. So we can see that actually A, B and C are very closely related because they share a common ancestor, whereas C is more distant compared to A and B there. So that's quite a detailed look at taxonomy. It's one of my, I've probably spent a little bit extra time there because one of my interests there. We're going to now look at another aspect of this topic, which is biodiversity. What actually is biodiversity and how we might measure it. Biodiversity is a measure of the diversity of life. It is actually comprised of three different areas. We look at the species diversity, so the range of species within a given habitat or globally, and there could be, in excess of 10 million species on the planet. I wouldn't be surprised if that's a gross underestimate. I suspect there will be well more species on this planet. So we've got our species diversity, the range of species within a given habitat. We also look at their genetic diversity, so the range of alleles within the individual populations that we find in a given habitat, and that was our last topic we looked at there. But also we need to think about the ecosystem diversity, the range of different types of ecosystem habitats that we have. Generally speaking, if an environment has a wide range of different ecosystems, then that's going to encourage an increase in species diversity and hopefully genetic diversity, so having populations that have a wide range of alleles in them, especially if you've got lots of different habitats, and you might say have a bird species that is moving between different habitats. That might actually encourage genetic diversity because there might be selection in this habitat for this particular allele, and there might be different type of selection in that habitat, and that might drive up the genetic diversity. So that's the idea of biodiversity. It is very important because we are dependent on the natural world. I think humans forget this very often, that we're kind of some all-powerful species that we can make and destroy anything we like. But irrespective of that, we depend on living things for our very survival. The most obvious reason is obviously plants. They're producing all of our food in our food chains. They are producing all the oxygen in our atmosphere that we require for aerobic respiration that is keeping us alive right now. But other organisms are playing important roles within our lives without us even realising it. Microorganisms are recycling dead organic matter, that helps break down our sewage and our waste and things like that. They help to recycle elements in the soil to help our crops grow. Bees and other pollinators are enabling our crops to grow. We cannot exist without the natural world. It's impossible. And we need to protect it, because as soon as biodiversity starts to drop in a given place, that ecosystem can then really start to become unstable, because if you've got a very low diversity environment, removing a certain number of species, maybe there's been extinction or a change due to climate change, removing a few key species will have a catastrophic effect on a low biodiversity system, because they might play a key role. They might be a keystone species. Removing them will affect everything else in the food web. But if you've got a very biodiverse environment, removing one or two species might have less big impact, because other species will take up the slack, if you like. So it's very, very important that we maintain and consider our biodiversity. But how do we go about measuring something like biodiversity? And that's where we look at this idea of index of diversity. So I'm just going to write up the equation and I'm going to talk my way through it. Just before I talk about this old equation here, cheeky cup of tea, I think it's in order. Lovely jubbly. All right then, index of diversity. Now, this equation might seem intimidating, but actually it's not too bad once you get used to having, practicing a few of these. They're not too bad. When you're looking at the diversity within a habitat, you've got to factor in two things. The species richness, so the number of different species you would find in a given habitat. But also you need to think about the number of individuals of each species in a given habitat. So you might have two habitats, A and B, let's call them, and they may well have the same number of species. They have the same richness. You think, oh yeah, they're just as diverse as each other. But if you were to count the number of individuals of each, you might see that one, let's call it A, has got a large number of each species. So it's got what we call a good evenness. Whereas habitat B might actually be the opposite. So it has the same richness, same number of species, but we might find that one or possibly two have large numbers of individuals, but fewer numbers of the others. And actually we would see that habitat B is less diverse overall because it has a lower evenness. The distribution of individuals of each species is is not as balanced. And actually those sorts of habitats are the ones that are vulnerable to any kind of change in the environment, because like I said before, if you affect one of those really kind of abundant species, it can have a huge knock-on effect onto the others. So how do you go about using this equation? Let's say I've found three species, A, B and C. So big N stands for the total number of organisms of all species. So what you'd have to do is look at the number of organisms that we have. So let's say for A it's 5, for B it's 4 and C there's 3. So they're the number of individuals we counted for each of those species. To work out big N, you would add them together. So we would have 12 here is our total, and that gives you the big N. But to actually work out the value that is in the top of this equation, you would have to do that number multiplied by itself minus 1. So our value here is 11. So 12 times 11 would give you that top value there, okay? To work out this lower sequence, well, we're looking at this little n, which is the total number of individuals of each species. So in the case of species A, that is 5. But we've got to do this whole equation down here. So this symbol here means sum of, they're adding them together, of little n multiplied by itself minus 1. So what I tend to do is do this next to each of them. So in the case here is, I've got 5 times by itself minus 1, which would be 4. So 4 times 3, and then 3 times 2, like that. Okay. I would then add up these values here, and that would give me that value then. So I would use the answer from this one to go here, my answer from that row to go there. Okay. And what I might do is I might cut to an example where I've actually completed it, so you can see how it all works. Okay, good. So in this example, I have got myself 5 different species. To keep things simple, I've counted the same number of individuals of each species. So actually we can see that this population is very even in that respect. And let's work our way through it. So to work out big N, you would just add up the number of individuals you have of each species, which gives me a value of 50. I would then multiply it by itself minus 1, which is 49. And that gives me that value there. Okay, and then to work out the lower part of the equation, the little n part if you like, I've multiplied each number by itself minus 1, so that gives me 10 times 9, which gives me 90. And because all of my each of my species has the same number, I end up with all those 90s, and then I just sum them all, add them all together, and that gives me 450. And then to finish off the equation, you would just take that big N value we got there, or at least that part at the top of the equation, and divide it by the 450, which gives us the value of 5.44. Okay, and that's the index of diversity. That's what you've gone through that hard work to calculate. The larger that number, the more diverse your habitat is, and therefore you can make a comparison between another habitat. And if that number was smaller in another habitat, we can assume that it's less diverse. But always take into account the richness and also the number of individuals of each species. That is very, very important. Okay, so the next part of this topic to look at is how agriculture, farming, has impacted on biodiversity, and some of the methods that we could do with our farming practices to increase biodiversity. Okay, if you were to think about a crop field, what would you see? Well, you would probably, let's say something like wheat. You would see that you've got one species there, a monoculture as we call it, all one grass species. There might be the odd weed here and there, but generally speaking it's all one species. So our species diversity is going to be very, very low in your average crop field. What about the genetic diversity of our crop? Well, we've selectively bred it to have uniform characteristics. Often all the individuals are probably clones of each other, so they're actually going to have incredibly low levels of genetic diversity because we want to ensure our crop is going to be exactly what we want it to be. And in terms of habitat diversity, well, that original patch of land, there might have been a bit of woodland, there might have been a pond here, a bit more open grassland, whatever it may be, a range of habitats. Well, we've got rid of all that. We've drained the ponds, we've cut down the hedgerows, the trees, ploughed the land, so it's uniform. So in terms of agriculture, it has a really devastating impact on biodiversity because it lowers species diversity, genetic diversity, and habitat diversity. So if you've ever been on a plane, you really would have noticed just how much agriculture covers the UK. You just see this patchwork of fields between our urban centres. So a huge swathe of the country and globally has been given over to agriculture, which are effectively biodiversity deserts. Now, it's not all bad, and I appreciate that agriculture is very, very important because it's providing us all with food, and we need to maximise our food production, but we need to do so sustainably and also keeping biodiversity in mind, because if we continue with this very intensive farming that we're doing, we're going to find that in the future we're going to have some big problems because it's not going to be sustainable, and actually that we might start seeing biodiversity crashes, and a lot of species are actually directly or indirectly allowing our crops to grow, and if they're affected we're not going to be able to produce any food anyway. So we've got to really think about what we can do with biodiversity. So here's a few ideas that what we could do to have food production but maintain that biodiversity at the same time. So plant hedgerows around the field, because that's going to provide more plant species. It's going to up that plant species diversity, and that in turn is then going to provide more habitat and more food sources for other species like birds and insects. So often that's quite a common point. Increasing the plant diversity will increase insect and bird diversity because the plants provide more food and more habitats. We could maybe leave certain patches of the field maybe not as well drained, allow ponds and things to develop at maybe a corner of one of the fields. Again, that's going to increase our ecosystem diversity in there. We could do, instead of using lots of inorganic fertilisers, which are great for yields, but they have huge environmental problems like eutrophication, why not do something like crop rotation, where you are alternating your crops each year with a legume, which is a nitrogen fixing crop like peas or beans. They'll actually increase the fertility of the soil because they can fix nitrogen, which will over time increase the nitrate concentration in the soil. And also by rotating it, it will actually help control pest populations without having to use pests as well. We could do what a lot of ancient cultures did in the past of intercropping. So actually having multiple species of plant growing in the same fields. The Aztecs were, and other South American cultures are quite famous for having maize, squash and beans within the same field. And that way you would get a good range of food sources, have a higher species diversity, and actually those plants would actually support each other. So the beans are legumes, they're actually increasing the nitrate content in the soil. The corn acts as a kind of upright to help the squash to grow and so forth. So actually they all worked quite well in harmony. So we could do something like that rather than having these big monocultures. Another thing to consider is possibly leaving certain areas to be set aside, encourage native tree species to grow, because again they're going to provide more habitats and move more food sources for other species that are very important. And if we do things like that, we might lower our productivity of our fields, yes, I can see that being a point, but we will actually make those fields and that food production sustainable for the long term, so we can actually keep feeding our species well into the future, rather than running our planet into the ground, which is probable if we carry on doing what we're doing. Okay? And the last little bit we're going to look at in this topic is the concept of sampling. How do we go out into an environment and work out the diversity, the distribution of species within a habitat. So I'm only going to touch on a little bit on this part of this topic because you do a lot more in year 13 on it. But sampling is, you know, very important because it helps save time. If you were to go out and count individuals of every species within a habitat, you wouldn't have time. It wouldn't be practical, particularly if you've got a very large and expansive habitat. So sampling will save you time. But you've got to do it in the right way. First of all, you've got to do it randomly. You've got to find some method of sampling your habitat without any kind of bias coming into place. So some of the obvious things, using like a random number generator to place your quadrat on, which is obviously the quadrat is that square grid that we can use to count the number of species. Not all quadrats have to be that size. If you were looking at a very, very large patch of forest, then you might even do like hectare-sized quadrats, for example. It depends on what's appropriate. So you definitely do random sampling because it removes any potential bias from the results. You've then got to figure out what is the optimum number of samples to take, quadrats to place, if you will. The larger the number you do, the more representative, the closer it will be to the real value. The fewer you do, the less representative it becomes, but the quicker the process becomes. So you've got to find this sort of middle ground where you have selected the optimum number of quadrats. I would suggest in the exam question a minimum of 10. So place a minimum of 10 quadrats if you want to have to place a value with it. With that, then you could then start to count the number of individuals of each species you're collecting the percentage cover, and with that you could, you know, calculate something like an index of diversity. And that's it. That's the topic. That's the majority of the AS course covered in these videos, at least 50% of it. And next week we'll start looking at some year 13 content, and we'll start looking at photosynthesis, which I think you're going to find hopefully very interesting, but certainly very challenging too. And I'm going to break that down into lots of shorter videos. So this is Dr. P signing off. Stay safe, look after yourselves, look after your families, have a great day. Cheerio.